I'll just wait until the slides are off. Perfect. So my name is Merel van der Thiel, and as was so kindly introduced, I'm from Maastricht University. And all of us here are part of the Human Brain Mapping Organization. Very excited that you're all here for the first day of this conference, but I would like to put the emphasis on the human part. So we're all interested in imaging the human brain. So I am here to remind you today that the brain that you are scanning is actually inside of a human being. So I have nothing to disclose. However, I would like to acknowledge that part of this presentation I have to credit Molly Bright, who normally gave the introduction of this talk, but she currently couldn't be here because she has very important other matters to attend to at the moment, which is her little daughter, Zoe. So she has to take care of her. But I would like to acknowledge her because a large content of this presentation is due to her. So, as mentioned, we are all interested in the brain to study, for instance, neural activity or functional connectivity or correlates of behavior and cognition. So we are studying this inside of the brain, and this brain is inside of a head. And this head is attached to the body. So we are already quite used to thinking about things that might influence the data that we are acquiring. For instance, did this human that we're scanning have a large meal? Are they very caffeinated? Did they sleep well? Are they stressed? Did they do a lot of exercise right before the scan? Or do they exercise a lot in general? Do they smoke? How much do they smoke? Do they have anxiety, or is there any discomfort during the scan or general discomfort? So these are measures that we normally take into account, and we try to correct for these as well. But there are some measures that we cannot really correct for. So we are measuring the brain inside of a human being who's inside the scanner, who is breathing. And this also causes some signals. So how do we correct for those? For instance, by measuring them, by collecting them, and then we can use it to model and filter out. So today I'm going to talk about what different signals in the human brain, in the body, could affect the data that you're collecting in the human brain. So I will talk about the lungs, the heart, and other parts of the human body. For instance, the skin, the eyes, and the guts. Then I will go a little bit into the waste clearance system of the brain, which is thought to be driven by respiration and heart rate, and the CSF, the cerebral spinal fluid, plays a large part into this. And lastly, I will talk about the autonomic nervous system, which is the system that drives all of these physiological measures. So let's start with the lungs. The lungs are inflating and deflating approximately 12 to 16 times per minute. And before we go into more detail, I would like to make an that's a difficult word, between two things. So you actually have the expansion of the chest, the movement, the thoracic movement, and the respiration. So when we talk about breathing, these are kind of combined in our conversation. But they are actually two different things. So chest expansion is really the movement of your lungs, the inhalation and the exhalation. So the lungs expand when you breathe in, and when you breathe out, they contract again. So respiration is mainly about gas exchange. So the oxygenation and so oxygen and CO2. So this really is focused on the thin membrane that separates the blood gases in the arteries from the gas in the alveoli. This requires a pressure gradient, so from the blood to the lungs. And oxygen then moves out of the inhaled air and then into the blood hemoglobin, and carbon dioxide goes out of the blood and is then subsequently inhaled. So that is what respiration specifically refers to. However, these two are very interlinked, so there might be two different definitions, but they definitely have an influence on one another. So how do we measure this, and how does this affect your fMRI signal? So breathing causes your head to move in a periodic manner. And as you can imagine, this is primarily in the Z translation. So you breathe in and you breathe out. Your head goes up and down. So this can cause misalignments and also spin history effects due to the motion. So this mainly relates to the chest expansion, as I said, the movement of your thorax, and this is an immediate effect. So it affects the volume that you're acquiring in that minute and also the ones after because of the spin history a little bit. Furthermore, there is an influence on the chest position that can influence your B0 field homogeneity. So for instance, this can cause ghosting artifacts and head motion or apparent head motion in the face encoding direction. This also is mainly caused by the chest expansion and is an immediate effect. Furthermore, this can influence your arterial blood gas levels. So that relates to the fact that CO2 has a vasodilatory response, which then has an effect on your vascular tone. So when your vessels dilate, your blood flow increases, that then has an effect on your fMRI signal. So this is mainly affected by respiration, by the gas exchange part, and it has a delayed effect because the vasodilation then subsequently needs to cause the increase in blood flow, and that will have a bit of a delayed effect. How do we measure this? So we mainly measure this by a respiratory belt. So this is a stretchy belt that you put around the body that then looks at the changes in the chest position. You can place the belt at multiple different places in the body. So here you can see 3 examples on the chest, the bottom rib, and the abdomen. You might ask, so which one is the best? This depends a little bit on different vendors and on your own study. It might be that some participants might be more compliant with one or the other location, or that your data might have a better signal depending on one or the other placement. Another thing to remember is that it might be very nice to actually place the belt when the subjects are already in a supine position, because when you sit down and then you go lie down, it might move a little bit, which then has an effect on the measurements. Furthermore, your breathing style can also evolve as the scan, can also change while the scan evolves. For instance, somebody might be more relaxed when they're in the scanner longer, which then influences their breathing as well. Another way to deal with this is to use multiple belts, if this is something that your participant is also comfortable with. So this is an example of the signal that you will be getting out. So it's like a sinusoidal wave. And you can also see an example, I don't know if it's very clear to see, in yellow, of the clipping of the signal. So that means that a part of the signal is not acquired. This could be because your respiratory belt is either too tight or too loose. We will talk a little bit more about that later. So how do you measure the respiration, the gases? We can do this with a nasal cannula. So you can measure the oxygen and the CO2 in your breath. This is a non-invasive surrogate of arterial sampling, because arterial sampling is a little bit difficult to do inside of an MRI scan. So this is a good alternative. It is like a plastic tube that kind of goes inside of your nose, and goes around the ears, and you tie it up your chin to make sure that it doesn't move as much. You need to extract the end tidal values. So here you can see an example of the signal from the nasal cannula. Oh, it got off a little bit. So on the top you can see the blue measurement of the CO2. So when somebody breathes out, the CO2 rises, and then you can see that there is a sharp cutoff, and that is the last part of breath of the lungs. So that's actually what you want, the end tidal values. Then when somebody breathes in again, you can see this steep decrease in CO2. And at the bottom, you see the poor end tidal estimates. This is a sinusoidal wave. You can't really see the in-breath that well, so that kind of shows that this signal is not very good. So you need to calibrate an identical setup between your different subjects, so the number of sample lines and the cannula, and also the vacuum settings. There is one backdraw. The cannula will always have some room air because it's not completely sealed off. And the participant needs to breathe through their nose. So for some people, they might be less compliant to do this. So another option that you could do is to use a mask, but that might be less comfortable. So that is something to consider. So now let's talk about the heart. The heart pumps approximately 60 to 100 times per minute. And once again, we have two different things. So when you think about the heartbeat and how this affects the fMRI signal, you might think about the heartbeat, the pumping of the heart. But another factor is the heart rate. So that's actually the frequency at what the heart pumps. So the heartbeat is the cardiac cycle, which involves the ventricles contracting during systole and relaxing during diastole. The contraction then pumps the blood through the circulatory system, and that the goal is to deliver oxygen and nutrients to the body through the bloodstream. And it is also to remove the waste products once again from the system. Then the heart rate, as I said, this is the rhythm, and that is set by pacemaker cells inside of the heart, which are affected by the autonomic nervous system. So the parasympathetic nerves slow down the heart rate, and the sympathetic nerves increase the heart rate. And these are in constant interplay with one another. And for instance, this could be triggered by physical activity, metabolic byproducts, and many other things, like stress, for instance. They all impact heart rate. I don't know if the video works, but how does this then affect your fMRI data? So the heartbeats creates a rapid traveling pressure wave through the vascular system. So the video doesn't work, but what you would normally see, you might have seen it already, which is good because then I don't need to show you the video, is this pulsating brain due to the heartbeat. This could move because it pulsates. This could lead to local movement artifacts and also to pulsatile flow artifacts. So within the voxel, the spins that then flow through, that might be different because of the spin history. How can you measure this? You can measure this using a PPG. And this is an optical measure of changing blood volume in the periphery. So it's done through light, so the LED illuminates the skin, and then there's a sensor that records the reflected or the transmitted light. This is typically done on either the finger or the earlobe, but it's good to keep this also consistent because this might have a different pulse wave travel time dependent on the location, and also the oxygenation dynamics and levels could vary. There's alternative recordings to the PPG, which are pressure-based sensors and electrocardiogram or ECG. And the ECG is often available in many scanner systems or many MRIs complexes. So Sarah will talk a little bit more about this on how to acquire data and then to clean this data right after. And then subsequently, Stefano will talk more about how to model the physiological driven signals that you get from these measurements. So what exactly is moving other than your blood? Well, your brain is within your skull, and your skull is non-compressible. It is thick. It cannot comply at all. So when the heart pumps and the blood goes into the brain, something needs to go out, because otherwise the pressure will build up and your head would literally explode. So what goes out is the cerebral spinal fluid, or the CSF. And this interchange between the blood and the CSF is coined the Monroe-Kelly doctrine, where blood goes in and then CSF goes out. And this concept has been used or has been applied in this novel field that I said before, which is the glymphatic system or the waste clearance system. Because cerebral spinal fluid plays a large role into this system, the Monroe-Kelly doctrine is very important for this as well. So I'll talk a little bit more about the waste clearance system, but let's start with the clearance of the body. So we learned in school that nutrients and oxygen get delivered to the body through the arterial system, and then the veins subsequently drain this back. And waste then gets cleared by the lymphatic system in the body. So how does this work in the brain? Well, we have an arterial system, we have a venous system, but the brain has no lymphatic system. So for many years, it remained unclear how this metabolic waste then got cleared from the brain. Until around 2015, when the waste clearance system of the brain got rediscovered. So how does this system work? You have the cerebral spinal fluid that surrounds the brain, which you can see here at number 1. Then this fluid dives into the brain through these spaces that surround the blood vessels. They're called perivascular spaces, as you can see here at number 2. And then at number 3, in the interstitial fluid, this fluid gets exchanged with the interstitial fluid, it picks up the waste products, and is then flushed out of the brain. So this is done on either the arterial or the venous side of the brain, the flushing out of the waste products. And that is something that there is a little bit of controversy on. So it could either be on the arterial side, which is coined the intramural periarterial drainage system, or IPAD, it's a bit easier to say, or on the venous side, to the venous perivascular spaces, which is called the glymphatic system. So I don't really know, or it's very unclear which one this is. So normally I personally refer to it as the cerebral waste clearance system, just to stay away from this distinction. But both are used in the literature. So it is important to know that both of these terms could be used if you search in literature and what the distinction exactly is. So why is it interesting for you, for people from OHBM, to look at the cerebral waste clearance system? Well, this system is, for instance, very important in Alzheimer's disease and other neurodegenerative diseases. So in Alzheimer's disease, you have amyloid beta and tau that are not properly cleared from the brain, which then causes these aggregates and leads to the disease. But this doesn't only happen in Alzheimer's disease. There's many proteinopathies that cause this aggregation of waste products that then lead to the disease. And many of these diseases are diseases that are being investigated here at OHBM. So it might be very interesting to you to additionally acquire information on this waste clearance next to your data that your either cognitive or behavioral data that you are acquiring. And one way to do this is, for instance, to look at the cerebral spinal fluid measures, which is the first part of the waste clearance system. So there is a way to look at this simultaneously with your fMRI acquisition, but this is something that Laura will talk about more later this afternoon. In addition to the CSF flow, you can also get information on the drivers of the waste clearance system. So this is considered to be multiple drivers that all interact together, such as cardiovascular pulsatility, respiration, and the vasomotion, the spontaneous constriction and dilation of your vessels. So these are considered to drive the fluid motion of the waste clearance system. And thereby, if you acquire your physiological data, you can also gain information on the drivers, or the considered to be drivers of this waste clearance system. So let's take a step back and talk about the rest of the body, the physiological signals that you can collect from the rest of the body. I will start by talking about the skin, the largest organ of our body. So skin conductance, you can measure this using skin conductance, which is also called the galvanic skin response. And you can measure this using electrodermal activity. So your skin conductance response is proportional to the number of activated sweat glands in your skin. And these activation could be, for instance, through stress or arousal or emotional excitement. And that could be in two things: in a response to a stimuli, so that's called an event-related skin conductance response, or a non-specific skin conductance response. You also have spontaneous low-frequency fluctuations. So these are not evoked. So the skin conductance response originally comes from the fight or flight response. So if you see something that's very threatening in evolutionary terms, you might have to run away from it. Your body gets ready to run, and it starts to cool itself down by sweating to prepare yourself for this activity. So this is something that is still triggered in a smaller way in our day-to-day life now. For instance, by thoughts and emotions, they can trigger this in a smaller effect. this is an example of the skin conductance data, so of the study. So first, they are given instructions, and you can see that there's a low response. Then there is, oh, that went a bit too fast. Then they engage in this task, like this gambling task, where they have to make risky choices. And then subsequently, there's a questionnaire, and you can see it going back down. So in addition to this tonic response, you also have these phasic responses, which you can see here with the stars. And these are, the phasic responses. All right, and these are considered to be a proxy for sympathetic nervous system activity. So how do you measure skin conductance? You first need a clean skin and to apply electrolyte gel and fix the electrodes. So as far as I understood, there's not really an established standard positioning, but if anybody disagrees, please let me know. But an example to do this is, for instance, on your fingers, which you can see here in the image. To be able to collect this, as with a lot of physiological data, you want to minimize the hand movement. That could be done by supporting the arm or the hand with cushions. And you can test whether you get a good skin conductance response by asking the person to take a deep breath outside of the scanner to evoke this skin conductance response. With the data that you then get out, you can use filtering and other signal processing techniques to separate the phasic from the tonic responses. And because you're measuring skin conductance, you have to be careful with the temperature, humidity, and hydration, and medication of the subject, because these can all influence your skin conductance response. So now we go to the next part, which is the eyes. So there's many MRI-compatible eye tracking systems out there, and these could be used, for instance, to look at where the visual attention has been focused. And this could be important if you have a very complex stimuli, for instance. But next to this, you can use it to measure your pupils through pupillometry, so the dilation of your pupils. So this responds to emotionally charged stimuli, both positive and negative. It also responds to a high cognitive load. So when you have a very high cognitive load, your pupil size also increases. Because you're looking at your pupils, you have to control the light levels, both in the room and from your stimuli, to make sure that your pupil response isn't just due to the luminescence of the stimuli. So there has been many studies and many papers that found that there is a relationship between negative visual and sensory motor bold signal changes with an increased diameter of the pupil. And there's also many other signal changes of bold that could be associated with the pupil diameter change. So let's touch upon the gut. This is a very emerging field using fMRI to study the gut-brain axis. So there's pacemaker cells in the stomach that generate a continuous rhythm, the gastric rhythm, of about 0.05 hertz, even when the stomach is empty, so not only when you're digesting. And you can measure this using electrogastrography. It's also finding very difficult to pronounce, but I will just call it EGG. So in this way, you can place electrodes on the skin to measure the rhythm of the stomach. And there has been studies that have found a significant relationship between the EGG signal and the bold fMRI changes. But Ignacio will talk about this more later during this course. And let's go now to the autonomic nervous system. So the autonomic nervous system consists of two parts: the parasympathetic nervous system, which is most active during rest, and the sympathetic nervous system, which is thought to be more active during activity and stress and arousal, for instance. So you can see here that we discussed all the different parts where I placed stars. We discussed all of the different physiological parameters to measure parts of this autonomic nervous system. So specifically, you can use pupillometry to look at the pupil dilation, which is more constricted during parasympathetic activity and is dilated using sympathetic activity. The airways, so the breathing, there's a more deep and intense breathing. You need more oxygen when your sympathetic nervous system is activated because you get ready to have your fight or flight response. You have an increased heart rate when your sympathetic nervous system is active. And we also talked about the sweat glands. So when your sympathetic nervous system is more active, you will also have more active sweat glands. And lastly, we talked about the EEG, which can measure the activity of the digestive system. So the autonomic nervous system altogether kind of regulates all the physiological parameters that we have shortly discussed and that we will discuss more today. But Pinar will talk more about the autonomic nervous system and the influences and what you can measure using fMRI. So to summarize, we talked about the lungs and the effects that this can have on your imaging data and also about the heart. And both of these together are thought to drive the waste clearance system, which we discussed that CSF plays a major role in. We talked about other physiological signals from the body, specifically about the skin, the eyes, and the gut. And then lastly, we talked about the autonomic nervous system, the regulator of your physiology, which consists of the parasympathetic nervous system and the sympathetic nervous system. So that concludes my talk. I would like to thank you all very much for your attention. I would like to thank my colleagues, and once again, Molly Bright for the content of this presentation. So my main specialty is imaging of the waste clearance system. So if you have any interest in that, or if you have any further questions, please feel free to talk to me afterwards as well. Okay. But if you have any questions now, you can also ask them, of course. Thank you.
I'm monitoring both the app, by the way, it's ohbm.cnf.io, if you want to ask questions there, and I'm monitoring Slack. How many of you managed to join Slack? Okay, at least somebody managed. Awesome. So we have one question on the app. I don't know if you want to take a little bit more time to ask that. I'm going to start here, and then we can relaunch it at the panel later. Are there any approaches to remove neural effects from breath-filled fMRI when used for cerebrovascular activity mapping?
That's a very good question. That is not necessarily my area of expertise, but I know that there will be some speakers coming after me, and I would highly suggest asking that question to them again. You will get a very more informed response.
Is there any other question? If you want to stand up. Oh, sorry, yeah.
I was just wondering if there is any study that was done about the influence of heat waves of women in menopause about fMRI response to stimuli or something like that.
Mm-hmm. That is very interesting. I personally am not familiar with the literature, but I think that that would be something very interesting to look at. I think there has been studies that have looked at the temperature of the brain and how this will affect your fMRI response. So I assume that cyclic changes would have an influence as well, and the menopause changes as well. Yeah, that's a very interesting question, yes.
Any other question? I'm just going to check the app if there's an update. Okay. So if there's no other questions, let's thank once again our speaker.
Thank you.